Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. We're going to continue our lecture today talking about protein synthesis. Last time I saw y'all, we talked about transcription, which is the first part of protein synthesis and happens in the nucleus. Today we're going to move into the second part of protein synthesis. It's known as translation, and as a reminder, this happens in the cytoplasm. Let's go ahead and hop into it and see what is going on here. So, protein synthesis, just as a reminder, another name that you might see for protein synthesis is known as the central dogma, okay? And this process, protein synthesis, it has two steps. The first step is known as transcription, and in transcription we go from DNA to messenger RNA, and that happens in the nucleus. The next part is known as translation, and that's step 2. In translation, we go from messenger RNA to protein, and this happens in the cytoplasm. Once the mRNA molecule is written, it is translated into proteins, okay? So, in protein synthesis, remember that this occurs in the cytoplasm, right? That translation, actually making the proteins, all happens in the cytoplasm. And this occurs in 3 steps. These 3 steps should look familiar because they are the same steps in DNA replication and in transcription. The first part of protein synthesis, the first part of translation, is the initiation phase. What's going on here is that mRNA is going to bind to rRNA. The rRNA finds the start codon, okay? And again, let me just go through these different steps and then I'll show you a picture of what's going on here in just a second, okay? So once the rRNA and the mRNA are connected, we're going to start the next phase, which is known as elongation. What happens here is that tRNA is going to bind to the rRNA and it's going to start dropping off amino acids. It knows which amino acids to use because the ribosome is reading the mRNA in sets of 3s. Those are called codons. A codon is going to code for a specific amino acid. Okay? This happens until we get to the last phase, known as termination. At a stop codon, the growing chain of amino acids is complete and we have made a polypeptide. At that point, the polypeptide needs to fold a little bit. The folding process for these are very complicated. Once it's done folding, we'll make a protein, okay? So let's get into some of the more nitty-gritty details here. Okay, so protein synthesis. What's going on here is that the RNA reads the mRNA in sets of 3 nucleotides at a time. 3 nucleotides is known as a codon, okay? Each codon codes for a specific amino acid. So real quick, just look at how similar those words are. Codon codes, right? That's a good way to remember the job of a codon. A tRNA carries the specified amino acid to the rRNA, okay? The tRNA uses its set of 3 nitrogen bases, known as an anticodon, to pair up correctly. So again, a codon and an anticodon go together. They are complementary. Because they're complementary, that's how we're assuring that we're using the right amino acid here, okay? The rRNA molecule builds the growing polypeptide chain 1 amino acid at a time. It's going to start linking those amino acids together. The bond between each amino acid is known as a peptide bond. That's why this thing that we're making before it's a protein, sometimes we'll call it a polypeptide, and that's because there's many peptide bonds in this structure. Lastly, the rRNA moves down to the next codon and continues the process. Okay, so let's look at that in a little bit more detail, what's really going on here. Okay, I'm going to walk y'all through it, point out some of the important things you need to know and how this whole process happens. Okay, so down here, this long line, this is the mRNA. Okay, we made this in the nucleus. So it's left the nucleus, it's come out to the cytoplasm. That's where these ribosomes are. Okay, so the mRNA is going to find a ribosome. At this point, the ribosome has been constructed. The large subunit, the lighter green, and the small subunit, the darker green, needs to come together to form a ribosome. So it's formed a ribosome. The mRNA is going to attach to it. Okay. What happens then is this ribosome starts reading this mRNA molecule. It's looking for something called a start codon. The same way when we were doing transcription, we had something known as a promoter sequence. Here it's called a start codon, and that's basically telling the RNA molecule where to start this building process. So it's going to read down this mRNA until it finds a start codon. Once it finds a start codon, it's going to start finding tRNAs that are complementary. These blue structures over here are known as tRNA, which the T stands for transfer. What's going on here is their job is to transfer these amino acids. The little circles here are amino acids. So their job is that they transfer the amino acids to the ribosome. Okay? How do we know what amino acid to use? Well, we use the codons. Every 3 nitrogen bases on the mRNA is called a codon. When it finds a tRNA that has a complementary base pair, that's how we know that we're using the right amino acid. Down here, 3 nitrogen bases is called a codon. On the tRNA, they're called anticodons. The way I remember that is that the word anti has a T in it, and tRNA has a T in it. So that's kind of an easy way to remember where are the codons, where are the anticodons. The anticodons are on the tRNA. Okay? So once we find those complementary base pairs, the tRNA is going to let go of its amino acid. See how this one doesn't have a little circle up top? That's because it's already dropped off its amino acid. The ribosome's job is to connect all of these amino acids together, and it does that by using something called a peptide bond. So here, what this structure is called, over here, it's labeled as a newly born protein. I would actually call this a polypeptide. Okay, this is a polypeptide. It's a structure with many peptide bonds holding it together. Okay, then what happens is you can follow these arrows here. The tRNA leaves the ribosome. This whole molecule moves down 3 more bases. The rRNA reads the next set of codons, and then the next tRNA comes to it, drops off its amino acid. So in this way, this is how we're building the proteins, because we're reading each set of nitrogen bases 3 sets at a time. That's a codon, and they're coding for these amino acids. Once this whole polypeptide has been made, it knows when to stop because it'll continue reading until we get to a stop codon. Then this polypeptide will leave the ribosome, and it's going to start going through this folding process. And once it's done folding, then we will have a protein. Okay, so I'm going to go through a couple more basic mechanics, and then we'll look at another picture that's kind of similar, and I'll walk y'all through it again. Okay, I know this stuff is confusing. The best way to learn it is just to look at these pictures and kind of talk our way through it. Okay, so let's get into some basic mechanics behind this whole translation process. Okay, so what's going on here is that we're adding 1 amino acid at a time to the growing polypeptide chain. This is done by the ribosomes, which, remember, the ribosomes are made up of rRNA. Okay, the ribosomes are made up of 2 parts, a large and a small subunit. They have to come together to make the ribosome really function. These 2 subunits come together before the translation process starts. Okay, the correct amino acid is specified by the nucleotides on the mRNA. 3 nucleotides on the mRNA is known as a codon, and those codons are coding for specific amino acids. At the rRNA, a codon on the mRNA and an anticodon on the tRNA meet. Okay, I know that's a lot of, it's like alphabet soup, right? It's a lot of abbreviation. What's going on here is that a codon and an anticodon, if they're complementary, they're going to match up. At that point, the tRNA drops off its amino acid, and the ribosome binds to the amino acid together using those peptide bonds. So let's look at another picture of this whole process here in just a second, and I think it'll clear it up a little bit. Here are some more basic mechanisms behind this process. So each amino acid in the growing polypeptide is specified by a codon on the mRNA molecule. This is how nucleotides in the mRNA codes for proteins. The ribosome translates the mRNA by reading the codons. Each tRNA molecule has an anticodon that pairs with the codon and an amino acid. When the anticodon and the codon match, amino acid is dropped off. The ribosome binds these amino acids together using peptide bonds. Let's look at this picture, kind of talk our way through it, and hopefully that'll clear up some confusion. So again, I'll kind of point some things out to you, and hopefully that'll clear up kind of what's going on here. Okay, so let's look at this long black line here. Okay, what is this? This long black line is known as messenger RNA. This is a copy of DNA. This was made in the nucleus, okay, through that process known as transcription. Once it's made, it leaves the nucleus and it comes out into the cytoplasm. So all of this is happening in the cytoplasm. As a reminder, this whole process that we're talking through is known as translation, okay? Is known as translation, okay? So what happens is this messenger RNA is going to meet up with a ribosome. This ribosome is made up of rRNA, okay? It's going to start reading this mRNA 3 bases at a time. And it's looking for something called a start codon. That tells us when to begin the building process. Once it finds a start codon, we're going to continue reading this mRNA 3 bases at a time. We're reading it amino acid by, we're reading it codon by codon, okay? Every time it reads a codon, that's telling the ribosome a specific amino acid we need. What's happening here is that the ribosome is reading it 3 bases at a time. It's trying to find, these are tRNA molecules, okay? Notice that on a tRNA molecule at the bottom you have 3 nitrogen bases, okay? That's known as an anticodon. And at the top there's an amino acid. That's what that little circle stands for, represents, okay? So when an anticodon and a codon are complementary, that's telling the ribosome that, hey, we've got the correct amino acid here, okay? When that happens, the tRNA is going to drop off its amino acid. The ribosome will then bond those amino acids together using a peptide bond. Then the tRNA leaves, the mRNA moves down 3 more nitrogen bases, the ribosome reads the next codon, and that whole process starts over again, okay? And so what we have here, look how many codons we've read. Each of these circles represents a different codon. And every codon that is read is coding for a specific amino acid, and the ribosome's job is to bond all those amino acids together using a special type of bond known as a peptide bond. At this point, this long chain is known as a polypeptide because there's a lot of peptide bonds there. Then the ribosome will continue reading the mRNA until we get to a stop codon. That tells the ribosome we're done. At that point, this polypeptide would leave this process, and the polypeptide would begin to fold, and once it's done folding, that's when we make a protein. Okay? So y'all, thank you so much for following along. I hope I cleared up some of that confusion. Your teacher has some other work for you to practice on, so make sure to give that your best effort, and I hope everybody has a great day.